Welcome to the video lecture for Unit 3, Module 7.3. In this one, we're going to discuss the lungs. As always, here are your learning objectives, so be sure to reference these when you're studying for both the lecture quiz as well as the unit exam. Let's go ahead and get started. So the left and right lungs are surrounded by the left and right pleural cavities, respectively. Each lung is a blunt cone. Its tip or apex points superiorly. The apex on each side extends superior to the first rib. The broad, concave inferior portion, or base of each lung, rests on the superior surface of the diaphragm. The lungs have distinct lobes that are separated by deep fissures. The right lung has 3 lobes, the superior, the middle, and the inferior, separated by the horizontal and oblique fissures. The left lung has only 2 lobes, the superior and the inferior, separated by the oblique fissure. The right lung is broader than the left because most of the heart and great vessels project into the left thoracic cavity. However, the left lung is longer than the right lung because of the diaphragm rises on the right side to accommodate the mass of the liver. The heart is located to the left of the midline, so its corresponding impression is larger in the left lung than in the right. In anterior view, the medial edge of the right lung forms a vertical line, but the medial margin of the left lung is indented at the cardiac notch. The primary bronchi and their branches form the bronchial tree. Because of the left and right primary bronchi are outside the lungs, they are called the extrapulmonary bronchi. As the primary bronchi enter the lungs, they divide to form the smaller passageways. These branches within the lungs are collectively called the intrapulmonary bronchi. Each primary bronchus divides to form the secondary bronchi, also known as the lobar bronchi. In each lung, one secondary bronchus goes into each lobe, so the right lung has 3 secondary bronchi, and the left lung has 2. In each lung, the secondary bronchi branch to form tertiary bronchi or the segmental bronchi. The branching pattern differs between the 2 lungs, but each of the tertiary bronchus ultimately supplies air to a single bronchiopulmonary segment, a specific region of one lung. The right lung has 10 bronchiopulmonary segments. During development, the left lung also has 10 segments, but adjacent tertiary bronchi fuse, generally reducing the number to 8 or 9. The walls of the primary, secondary, and tertiary bronchi contain progressively less cartilages. In the secondary and tertiary bronchi, the cartilage form plates arranged around the lumen. These cartilages serve the same structural purpose as the rings of the cartilage in the trachea and the primary bronchi. As the amount of cartilage decreases, the amount of smooth muscle increases. With less cartilaginous support, the amount of tension in those smooth muscles has greater effect on a bronchial diameter and the resistance to airflow. During a respiratory infection, the bronchi and bronchioles can become inflamed and constricted, increasing the resistance. In this condition, called bronchitis, the individual has difficulty breathing. Each tertiary bronchi branches several times within a bronchopulmonary segment, forming many bronchioles. These bronchioles then branch into the finest conducting branches, called the terminal bronchioles. Nearly 6,500 terminal bronchioles arise from each tertiary bronchus. The lumen of each terminal bronchiole has a diameter of about 0.3 to 0.5 millimeters. The walls of the bronchiole lack cartilage but are dominated by smooth muscle tissue. In functional terms, bronchioles are to the respiratory system what arterioles are to the cardiovascular system. Changes in the diameter of the bronchioles control the resistance to airflow and the distribution of air into the lungs. The autonomic nervous system controls the diameter of the bronchioles. It does so by regulating the activity in the smooth muscle layer. Sympathetic activation leads to bronchodilation, the enlargement of the diameter of the airway. Parasympathetic stimulation leads to bronchoconstriction, a reduction in the diameter of the airway. Bronchoconstriction also takes place during allergic reactions such as anaphylaxis in response to histamine released by activated mast cells and basophils. Bronchodilation and bronchoconstriction are ways of adjusting the resistance to airflow. These actions direct the airflow towards or away from specific portions of the respiratory exchange surfaces. Tension in the smooth muscle commonly causes the bronchial mucosa to form a series of folds that limits airflow. Excessive stimulation, as in asthma, can almost completely prevent airflow along the terminal bronchioles. The connective tissue of the root of each lung extended into the lung's parenchyma, or functional cells. These fibrous partitions, or trabeculae, contain elastic fibers, smooth muscles, and lymphatic vessels. The trabeculae branch repeatedly, dividing the lobes into even smaller compartments. The branches of the conducting passageways, pulmonary vessels, and nerves of the lungs follow these trabeculae. The finest partitions, or the interlobular septa, divide the lung into pulmonary lobules. Branches of the pulmonary arteries, pulmonary veins, and respiratory passageways supply each lobule. The connective tissues of the septa are, in turn, continuous with those of the visceral pleura, the serous membrane covering the lungs. Each terminal bronchiole delivers air to a single pulmonary lobule. Within the lobule, the terminal bronchiole branches to form several respiratory bronchioles. The respiratory bronchioles are the thinnest and most delicate branches of the bronchial tree. They deliver air to the gas exchange surfaces of the lungs. Before incoming air moves beyond the terminal bronchioles, it has been filtered and humidified. A cuboidal epithelium lines the terminal bronchioles and the respiratory bronchioles. They are only scattered cilia and no mucus cells or underlying mucus glands. If particulates or pathogens reach this part of the respiratory tract, there is little to prevent them from damaging the delicate exchange surfaces of the lungs. Respiratory bronchioles are connected to individual alveoli and to multiple alveoli along regions called alveolar ducts. Alveolar ducts end at alveolar sacs, common chambers connected to multiple individual alveoli. Each lung contains about 150 million alveoli. They give the lungs an open, spongy appearance. Each alveolus is associated with an extensive network of capillaries. A network of elastic fibers surrounds the capillaries. These fibers help maintain the relative positions of the alveoli and the respiratory bronchioles. When these fibers recoil during exhalation, they reduce in size of the alveoli and help push air out of the lungs. The alveolar epithelium consists mainly of simple squamous epithelium. The squamous epithelial cells, called type 1 pneumocytes or type 1 alveolar cells, are unusually thin and are the sites of gas diffusion. Roaming alveolar macrophages or dust cells patrol the epithelial surface. They phagocytize any particles that have eluded other defenses. Large type 2 pneumocytes, also called type 2 alveolar cells, are scattered among the squamous cells. Type 2 pneumocytes produce surfactant, an oily secretion containing phospholipids and proteins. They secrete surfactant onto the alveolar surfaces, where it forms a superficial coating over a thin layer of water. Surfactant plays a key role in keeping the alveoli open. It reduces surface tension in the liquid coating of the alveolar surface. Recall from 168 that the surface tension results from an attraction between water molecules at an air-water boundary. Surface tension creates a barrier that keeps small objects from entering the water, but it also tends to collapse small air bubbles. Without surfactant, the surface tension would collapse the alveoli much in the same way. Surfactant forms a thin surface layer that interacts with the water molecules, reducing the surface tension and keeping the alveoli open. If type 2 pneumocytes producing inadequate amounts of surfactant due to injury or genetic abnormalities, respiration becomes difficult. The alveoli collapse after each exhalation. With each breath, the inhalation must be forceful enough to pop open the alveoli. A person without enough surfactant is soon exhausted by the effort of inflating and deflating the lungs. This condition is called respiratory distress syndrome. Gas exchange occurs across the respiratory membrane of the alveoli. The respiratory membrane has three layers. It contains 1, the squamous epithelial cells lining the alveolus, 2, the endothelial cells lining the adjacent capillary, and 3, the fused basement membranes that lie between the alveolar and endothelial cells. At the respiratory membrane, only a very short distance separates the alveolar air from blood. The total distance can be as little as 0.1 microns, but it averages about 0.5 microns. Diffusion proceeds very rapidly across the respiratory membrane because the distance is short and both oxygen and carbon dioxide are small, lipid-soluble molecules. The plasma membranes of the epithelial and endothelial cells do not prevent oxygen and carbon dioxide from moving between blood and alveolar air. Certain diseases can compromise the function of the respiratory membrane. Pneumonia develops from an infection or any other stimulus that causes inflammation of the lobules of the lung. As inflammation occurs, fluid leak into the alveoli. The respiratory bronchioles swell, narrowing passageways and restricting the passage of air. Respiratory function deteriorates as well. When bacteria are involved, they are generally types that normally inhabit the mouth and the pharynx, but have managed to evade the respiratory defenses. Pneumonia becomes more likely when the respiratory defenses have already been compromised by other factors. Such factors include epithelial damage from smoking and the breakdown of the immune system in AIDS. The respiratory defenses of healthy individuals prevent infections and tissue damage, but the breakdowns of those defenses in AIDS can lead to a massive, potentially fatal lung infection. The most common pneumonia that develops in individuals with AIDS results from infection by the fungus Pneumocytosis urosei. Two circuits nourish lung tissue. One supplies the respiratory portion of the lungs. The other perfuses the conducting portion. The respiratory exchange surfaces receive blood from arteries of the pulmonary circuit. The pulmonary arteries carry deoxygenated blood. They enter the lungs at the end of the hilum and branch with the bronchi as they approach the lobules. Each lobule receives an arteriole and a venule, and a network of capillaries surrounds each alveolus as part of the respiratory membrane. Oxygen-rich blood from the alveolar capillaries passes through the pulmonary venules and enters the pulmonary veins, which deliver the blood to the left atrium. In addition to providing for gas exchange, the endothelial cells of the alveolar capillaries are the primary source of angiotensin-converting enzyme, or ACE, which converts circulating angiotensin 1 to angiotensin 2. This enzyme plays an important role in regulating blood volume and blood pressure. The tissues of the conducting passageways of your lungs receive oxygen and nutrients from capillaries supplied by the bronchial arteries, which branch from the thoracic aorta. The venous blood from these bronchial capillaries may return to the heart by either the systemic or the pulmonary circuit. About 1/8 of the venous blood drained into the bronchial veins passes through the azygos, the hemiazygos, or the highest intercostal vein, and enters the superior vena cava. The large fraction of the venous blood enters the anastosomes and drains into the pulmonary vein. This venous blood dilutes the oxygenated alveolar blood within the pulmonary veins. Blood pressure in the pulmonary circuit is usually low. Systemic pressures in the pulmonary circuit are about 30 or less. With such pressures, pulmonary vessels can easily become blocked by small blood clots, fat masses, or air bubbles in the pulmonary arteries. Because the lungs receive the entire cardiac output, any such objects drifting in blood are likely to be trapped in the pulmonary arterial or capillary networks. Very small blood clots occasionally form in the venous system. These are usually trapped in the pulmonary capillary network, where they soon dissolve. Larger emboli are much more dangerous. The blockage of a branch of a pulmonary artery stops blood flow to a group of globules or alveoli. This condition is called pulmonary embolism. If a pulmonary embolism is in place for several hours, the alveoli will permanently collapse. If the blockage occurs in a major pulmonary vessel rather than a minor branch, pulmonary resistance increases. The resistance places extra strain on the right ventricle, which may be unable to maintain cardiac output, and congestive heart failure can result. The thoracic cavity has the shape of a broad cone. Its walls are the rib cage, and the muscular diaphragm forms its floor. The two pleural cavities are separated by the mediastinum. Each lung is surrounded by a single pleural cavity, which is lined by serous membrane called the pleura. The pleura consists of two layers, the parietal pleura and the visceral pleura. The parietal pleura covers the inner surface of the thoracic wall and extends over the diaphragm and the mediastinum. The visceral pleura covers the outer surfaces of the lungs, extending into the fissures between the lobes. Each pleural cavity actually represents a potential space rather than an open chamber because the parietal and visceral pleura are usually in the close contact. Both pleura secrete a small amount of pleural fluid. Pleural fluid forms a moist, slippery coating that provides lubrication. It reduces friction between the parietal and visceral surfaces as you breathe. Samples of pleural fluid obtained through a long needle inserted between the ribs are sometimes needed for diagnostic purposes. This sampling procedure is called thoracentesis. The extracted fluid is examined for bacteria, blood cells, or other abnormal components. In some diseases, the normal coating of the pleural fluid does not prevent friction between the pleural surfaces. The result is pain and pleural inflammation, a condition called pleurisy. When pleurisy develops, the secretion of a pleural fluid may be excessive or the inflamed pleura may adhere to one another, limiting the movement. In either case, breathing becomes difficult and prompt medical attention is required.